This Watershed Watch training module will walk you through completing the new data form that our volunteers will use to record observations and measurements for their stream sampling location. These results will then be entered into the organization's database for anyone to access. As just mentioned, the new data form enables you to enter your findings at the stream site. It will also be used to record information related to your use of the R-card method for analyzing E. coli concentrations. The form will be mirrored in an online data portal where you will enter your results. We ask that you take a photo of your completed form and upload it along with your results so that it can be used for cross-check verification of your data entry. You may also want to keep your paper copies in a folder for future reference. This is what the new form looks like. It is divided into various sections to help you work through the sampling procedure methodically and record your results. You may want to complete some of the first section before heading out to your sampling site. At the top of the form, you'll begin by entering basic information about samplers and the site location. This includes the first and last names of participating samplers and the number of adult and youth participants. You should write in the number of your sampling site, which will be issued once you set it up in the Watershed Watch database. The location of each site is mapped and tracked in the database using its geographic latitude and longitude coordinates. Once you've established the correct coordinates for your site, you do not need to record this information again. For clarification, here's a little more info about coordinates and how to determine what these numbers should be. Latitude ranges from -90 to 90 degrees, and in Kentucky, it should be somewhere between 36 and 39. Longitude values range from -180 to 180, and Kentucky values will begin with a number between 82 and 89. Google Maps is an easy way to find the lat-long information. You can open it on your phone or computer and select the location, either by clicking your cursor or holding your finger on the sampling site. A pop-up window with the location information will include the coordinates for you, as shown on this slide. Next, you will need to fill in the sampling date. Then write in the time at which you begin your sampling procedures at the stream. We also ask that you provide an estimate of the total number of minutes that you spent driving to your site, conducting your sampling, and traveling home from your site. This information helps Watershed Watch track volunteer contributions for grant reporting. Note that you shouldn't duplicate this information if you have multiple sites. Please just record the overall total for all sites and report it once when entering data for one of your sites. When you are at your sampling site, observe the weather and place a check mark next to the description that most appropriately describes the conditions. Also, place a check mark next to the rainfall amount that has occurred during the 48 hours before your sampling visit. There are various ways to determine rainfall, including a rain gauge or various websites. We like the Kentucky Mesonet site. Here is the web address and precipitation page from the Kentucky Mesonet's website. This network of automated weather monitoring stations covers most of the state. You can click on the county location nearest to your site to see rainfall amounts over various periods, including the past 48 hours. Next, you'll be using your senses to observe various qualities in the stream environment. First, take a look at the color of the water. Hopefully, it's clear. However, it may be one of the other colors listed here, or you have the option to add another color description. Mark which color is most appropriate. Then, sniff the air above the water. Again, hopefully there is no odor. If that's not the case, indicate which of the odor descriptions you seem to be smelling. Finally, take a look at the surface of the water and place a check mark next to any of the floating substances you may see. It's important to know that a sheen on the water can be natural due to the breakdown of organic materials such as leaves, grass, or sticks. or from bacteria that grows in iron-rich waters. If you disturb the sheen by poking it with a stick or throwing a rock into it and it breaks apart into clumps, it is most likely natural. However, if it quickly reforms, it is more likely due to a petroleum spill or runoff. A telltale petroleum odor will also likely be a giveaway. Similarly, observed suds or foam can be naturally formed when decomposition is occurring. Natural suds will be white or light brown and have an earthy odor. They are usually found in slow-moving water or eddies, near submerged logs, or in large clumps floating downstream. If the suds are being caused by human activities such as a sewer system, yard fertilization, or detergent use, they will be bright white and fragrant and accumulate near the source. Sewage-related foam will obviously have a foul odor. Moving on to stream flow, you will estimate water level in the stream in comparison to the banks on either side. Using this graphic as a reference, note whether it is dry with no water, ponded with separate distinct pools of water, low with minimal connected flow, normal within the formed banks that naturally contain the stream flow, bank full over those natural banks, or flooded and spreading over the land adjacent to the stream banks. Estimating flow will get easier over time as you visit the stream repeatedly and become more familiar with typical flow levels. Please do not sample during flood levels for safety reasons. If the flow is bank full and levels are above your knees, please use caution and consider not sampling. And if the flow is ponded, you shouldn't sample because the results will not be representative of stream flow. Obviously, if the stream is dry, you can't sample. You should note information about trash and litter for potential later cleanup activities. Estimate the amount using the options provided. Turbidity refers to the clarity of the water, which can be affected by suspended soil, algae, or other particles. If the water is very turbid, it will be opaque and look similar to chocolate milk if it's caused by suspended soil or sediments. If you have access to an electronic turbidimeter, which measures turbidity in NTU units, you can collect and report that data. Or if you have a transparency or turbidity tube, you can perform the procedure using that device and report your result in centimeters. Next, you can take pictures of the upstream and downstream views from your sampling location, as well as photos of any notable stream impacts or conditions. These can be possible threats to the stream or activities that are being done to help protect it. When you enter your data, you will have the ability to upload these photos to the database. Here are some examples of upstream, downstream, and potential stream influence photos. Cattle in the stream can contribute to turbidity measurements, E. coli concentration, and possibly your field chemistry measurements. They can also affect habitat and biological conditions. Next, using the procedures learned in the field chemistry training, take and record measurements for water temperature, pH, dissolved oxygen, and conductivity. If you have access to electronic water monitoring equipment and are using it to take any of these measurements, note the type of equipment and confirm that it was calibrated prior to its use. The next section of the form relates to the incubation and counting steps of using the R-card method for your E. coli analysis. Following the guidance you received in the training, first record the time you place the cards in the incubator. Then, after incubating the cards for 20 to 24 hours, take the cards out of the incubator and record the time. For each of the 3 cards taken for this site record, record the number of E. coli colonies counted on the card, the water sample volume you used, which is most likely 1 milliliter, and multiply by 100 for each sample, A, B, and C. Then you'll calculate the average of these 3 results by totaling them and dividing by 3. You will record this average on the card where noted. Using your knowledge of the sampling area and observations while you are there, place a check mark next to any possible sources of E. coli to the stream. Contributions from waterfowl waste, such as those from ducks or geese, are common in parks or near ponds. Human sources can be anywhere homes are nearby in both rural and urban settings. Livestock sources can be from pasture land or feedlots. Pet waste is likely most concentrated in park or subdivision areas, and wildlife inputs may be more likely in forested areas. Finally, please write in the initials of the person who counted the E. coli colonies on the R cards. At the bottom of the data form, you can record any other observations or measurements that were not recorded above. Examples of noteworthy observations may include things like a suspicious pipe upstream with a sewage odor, tire tracks on the stream bank, excessive algae growth, cows in the creek, or dead fish on the stream bank. Or you could note any improvements along the creek, such as recent tree plantings or less litter. Once you've completed the information on the data form, you'll need to log into the Watershed Watch data portal and enter your results. The website address and QR code link are included on the form. You'll also be asked to upload a photo of your data form so that it can be used for cross-checking and verification of data entry. If you'd like, you can also keep your paper form for future reference.